Hello everyone, thank you for joining us today for our review of some of the key clinical data sets which were presented at the annual ASCO meeting this past weekend. We're going to be focusing today on the data that was presented for AstraZeneca's EGFR inhibitor Tagrisso in the Adora study, which was in adjuvant EGFR positive non-small cell lung cancer. We're going to be looking at the data that Roche presented for its anti-TIGIT antibody, and we're also going to be looking at the latest data for Johnson & Johnson's Karthi therapy in multiple myeloma. Before we talk about the data sets specifically, obviously, this year's ASCO meeting was slightly different. It was held in a virtual format as a result of the COVID-19 pandemic. We actually polled oncologists a couple of months ago to see how they anticipated that a virtual meeting would work. There's a link to that poll in the article where this video is embedded. We spoke to a few experts and a few attendees in the last few days. There was quite a consensus actually, which was quite interesting. I think the people we spoke to felt that there were benefits of it being held virtually in terms of access to presentations, being able to access the data and analyze the data when they wanted to. But the real downside that we heard was that the conversations that people have when they're meeting face to face, interestingly, the conversations that people sometimes have that aren't planned either, meeting someone they know or meeting an investigator, sometimes in a queue for a taxi or what have you, those conversations are really hard to replicate in a virtual setting. One of the experts we spoke to described it as 40% of the experience for 20% of the hassle, perfectly acceptable, but overall an inferior experience. There was also a bit of chat about how social media had kind of replicated some of those conversations. So Twitter in particular has been a really interesting platform to watch over the last few days. And one really interesting bit of feedback, which I think may become apparent over the coming months, is one of the experts we spoke to felt that being in a virtual format, it created a kind of a winner takes all situation, where the bigger data sets which were presented at this year's meeting maybe got a disproportionate amount of attention than they would normally, and data sets that were maybe kind of less high profile didn't get the attention that maybe they would typically deserve or get. So I think that's quite an interesting kind of take on it actually. We may try and survey physicians or oncologists in the next couple of weeks to see how their experience was. It would be really interesting to kind of find out whether they feel the virtual setup will change the way that data presented is then kind of impacts clinical practice. I don't know how easy it will be to capture that sentiment. I don't know how objective oncologists will be, but I think it's certainly something to watch. I think the conclusion would be though that ideally, these large medical meetings will be going back to their traditional format as soon as possible, and obviously we all kind of hope that happens as and when it does. So I'll introduce my colleagues. I've got Becky on the line, Michael on the line. Hi guys, how are you doing?
Great, good to be here.
Hi there.
So the three data sets I mentioned at the beginning we're gonna discuss today. The phase 3 Adora study, looking at AstraZeneca's Tagrisso, the phase 2 Cityscape study, which is looking at Roche's TIGIT antibody tiragolumab, and also some still early stage but really impressive data from the CARTITUDE-1 study, which is looking at Johnson & Johnson and Legend Biotech's BCMA targeted CAR-T treatment in heavily pre-treated multiple myeloma patients. So we'll kick off with the Adora study, and you know, is the Adora study going to be a game changer? A bit of background information: Tagrisso is widely recognized as the gold standard first-line treatment for metastatic EGFR mutated non-small cell lung cancer, and Adora supports its use in earlier stage patients whose tumors have been surgically removed. We knew before ASCO that the data was going to be pretty impressive. In April, AstraZeneca announced that the study was halted 2 years ahead of its completion date. And when the results were presented, the use of Tagrisso was shown to reduce the risk of the disease progression or death by 83% in stage 2 and 3A patients, which was the primary endpoint, and expanding that cohort to all patients enrolled in the study, which was down to earlier stage 1B patients. It was shown to reduce the risk of disease progression by a still impressive 79%. We can see, I put in a couple of charts that got a lot of attention over the weekend. This is the Kaplan Meier chart in the primary endpoint in the stage 2 and 3A patients. And looking at some of the comments that were made by the study presenter, the trial is a home run. It exceeded our expectations and was described in his concluding remarks as a practice-changing treatment for this patient population. And Richard Shilsky, who's the ASCO Chief Medical Officer, also, gave the study very, very positive feedback. I think the interesting thing about these results, based on some key opinion leader interviews that we've done in the last few days, I don't think there's much doubt that Tagrisso is going to be approved in this setting based on these data. We spoke to Dr. Jack West, who's based at the City of Hope Comprehensive Cancer Center. He's made a very passionate argument, I would say, on social media over the last few days about how he feels that although oncologists will quite quickly embrace Tagrisso in this setting and probably use it across the broad spectrum of patients who are identified before metastatic disease, he personally feels that really, in an ideal world, we would have overall survival data to see if actually Tagrisso provides is curative, provides a survival benefit in this setting. Interestingly, to kind of complement his belief that oncologists will use it despite this issue, we spoke to another expert who actually said that even though she's still, obviously it's still unclear whether this does extend overall survival, she's actually already using it in the adjuvant setting in certain patients. So I think that kind of goes to show the scenario that is probably going to play out here. Very impressive data. There is a caveat over this overall survival benefit, which hopefully we'll get to see further down the line. But yeah, I mean, it really was. I would describe it as the kind of the headline grabbing data at this year's meeting. Which, to go back to that comment about the disproportionate attention that certain data sets are going to get, I think one of the people we spoke to said, AstraZeneca will certainly be hoping that doctors see this data and are impressed with it, basically.
Yeah, well, it was the belle of the ball, and as you said, probably gained an outsized amount of attention given the dynamic this year with the virtual ASCO. Yeah, so I think the question for me was coming out of this, so it's about the Adora study is a population of patients who are less advanced, than where people have been using Tagrisso. So, that means that the bar for toxicities is higher. So it's just going to be a matter of whether physicians, what their experience has been like with the drug so far, and whether how much they take into account financial toxicities, and whether whether the profile as it stands is good enough before they actually know whether it extends survival. That's, I think, the real question that we just don't know at this point.
Yeah, I mean, interestingly, to pick up on something you said there, Michael, I mean, I think toxicity has obviously been mentioned. You're right, lower risk patients, but it was interesting that Dr. West, who's based in the US, where to put it simply, we typically associate there being less of an impact of financial toxicity. That's one of the arguments that he's most passionate about, is that actually, you're potentially giving this drug to patients who may survive anyway, and it costs I think up to $250,000 a year. Is that should be a consideration that's made when physicians and patients choose to use this therapy. I think the other interesting thing is the study was designed to last 3 years. Obviously, they've stopped the study early because of how compelling the disease-free survival data is. I think AstraZeneca has said that they feel that patients will likely be treated in the adjuvant setting for 2 to 3 years. But Dr. West also makes the point that we could kind of see certain patients being treated almost indefinitely with this. And again, going back to that idea of is the alternative course of therapy to give patients chemotherapy, which is which is known to work, and does have a survival benefit in patients whose earlier stage tumors have been resected, and then you provide a drug like Tagrisso to people on first relapse, so you're not over treating patients who are potentially already cured. So I think I think this debate is probably going to run up until approval happens. I've seen a few comments over the last few days suggesting that even though we're talking about this caveat around overall survival data, I think the fact that the disease-free survival data was so striking, and knowing how the FDA operates at the moment with cancer approvals, we could potentially see this, and this isn't based on what any kind of company disclosure, but I think we could potentially see this on the market in the US relatively quickly, to be honest. And I'm sure that the proof will be in the pudding then, to see how quickly the sales kind of increase. And Tagrisso is already AstraZeneca's biggest selling drug. It's a really important growth driver for them. I think before we quickly move on to the next subject, they also had some really compelling data for their antibody drug conjugate in HER2, in different HER2 positive cancers, I think lung and gastric among them. So I think if we're looking at kind of company level stuff, I certainly think AstraZeneca came out of this year's meeting with quite a bit of momentum. They look in good shape, and they obviously they have been kind of delivering positive news and positive sales growth in the cancer market for the last few years. So that doesn't look like stopping anytime soon, to be absolutely honest. Yep. Let's maybe move on to the next data set. Michael, I don't know if you want to talk a little bit about tiragolumab. Becky, I know you've been looking at this as well. It was obviously kind of, even though it was a phase 2 study, it was pretty eagerly anticipated, because novel immuno-oncology agents are highly anticipated. People are desperate to see this data.
Yeah, I think we're always looking for something to add to PD1, PD-L1. I'll let you go first, Becky. I'll sort of add what I got from KOL. Maybe I'll add that on top of, whatever takeaways you have from this one.
Sure, sure. So I think, in addition to just being a novel mechanism in a place that just hasn't seen an effective novel mechanism in quite some time, a bunch of the anticipation for this data was just tied to, Roche going from effectively nothing to, 6 or 8 separate studies, all expanding this mechanism to combine TIGIT with PD1. So, of course, we already know that in this non-small cell lung cancer market, the entity to beat in all its forms is Keytruda, sort of across a breadth of different labels within the setting. And so, the big opportunity here that this new mechanism would be going for would be to sort of, wrest some control from Keytruda in the setting. But as we go through the data, it's sort of a kind of a hit and miss sort of readout in the sense that, particularly if you look at this PD-L1 interaction, while we definitively, see the synergy of a checkpoint inhibitor with, anything else in the immune landscape, there is a definite physiological effect there, that these two targets are 100% interacting, but having it be, as you can see here, pretty much entirely restricted to this high expressor PD-L1, I should say, population, isn't so ideal as Roche thinks about trying to expand this into settings like, a chemo combo, say, that would, have more prevalence in a low PD-L1 population. So Roche had a post-ASCO call to sort of, walk through some of these, sort of the ins and outs of why they might be seeing this effect. They've got, a reasonable mechanistic rationale for why they would expect to see, TIGIT to interact with PD-L1, and not necessarily some of the other, like the TIM3s, the other kind of common targets that pop up in these. But the long and short of it is, they are still looking really hard to try to find, other biomarkers that might help them sort of spread out the identity of their responsive population a bit.
Yeah, well, basically that's what the KOL, so the KOL said he was very encouraged by the results in PD-L1, high expressors, but with obviously the caveat that this is a phase 2 trial, need to show it in phase 3. He was very adamant that this is not strong enough for them to seek accelerated approval, which I don't think they have, suggested they are, but he was just like, yeah, it's just definitely not strong enough to do any sort of thing like that. But yeah, he's very interested in this being a useful tool, this, the Atezo or Tecentriq with tiragolumab for that PDL high expressors, and it would replace, if the phase 3 works out, it would basically replace Keytruda as a single agent. And that's it. It's based on the data, you can quite clearly see that, we're not talking about replacing Keytruda across the board, or even, in the PDL1, 1 through 49%. It's a smaller group, but obviously at least it's a step forward, which, people have been looking for a while.
It certainly seems to be a potential threat to Keytruda, doesn't it? Assuming obviously that the phase 3 data is kind of,
in that subgroup for sure, like in that subgroup, definitely. But outside of it, it doesn't really look like it.
Yeah, and it was interesting on their call also, there was lots of pressure on management to sort of lay out a timeline and or reasonable expectations for when they would start a chemo combo study in more of an all-comer population. And, the company, Roche was not willing to attribute timelines to, such a trial, but certainly would not say that they weren't going to do it.
Yeah, and I believe I believe from speaking to one of the KOLs this week that Merck is in the process of to make the point, Merck is developing its own anti-TIGIT antibody, and I kind of got the impression that they are priming a study with Keytruda and the TIGIT and chemotherapy. I mean, one of the other interesting, talking about these corporate presentations on the sidelines at ASCO, it's quite interesting when you listen to Merck management that they seem, for want of a better phrase, slightly less committed to this mechanism at the moment. But I think that, I think they're probably, a combination of playing their cards close to their chest and talking almost from a position of relative strength, with obviously, Keytruda is the dominant molecule for a reason. It really has delivered, it set a relatively high bar for other checkpoint inhibitors and indeed, other treatments to kind of meet. So I think there's going to be some really interesting kind of corporate dynamics played out over the next few years around this mechanism.
You know what? Just to finish that point, because I think you brought it up and it struck a chord. One of the KOL that I spoke with, he brought that up as well. The fact that Merck has a TIGIT, and it's hypothetical at this point, but there's sort of a positive sheen, if you will, around Keytruda, where everybody thinks Keytruda is the the greatest thing since sliced bread for cancer. And whether that perception is reality or not, who knows? But in the it could be interesting in the sense that if Keytruda and the TIGIT data, their TIGIT data, is just as good as Roche's, then it might be tough for Roche to to horn in on that market, because Merck has they are the 800-pound gorilla at this point. So yeah, so it'll be an interesting dynamic.
I think maybe the other interesting way to look at the tiragolumab data is, I kind of felt from talking to people this week that this is one of those areas where maybe expectations from the investor and the Wall Street angle are slightly disconnected to the expectations and the assessment of the data from an oncologist perspective. I think lots of people have made the point that this was this is phase 2, but it was randomized, and maybe a bit of a criticism that has been thrown at pharma companies in the last few years is that they've rushed these phase 2 uncontrolled studies through. They've got a bit of positive data and then they've gone into a phase 3 and we've seen it not meet expectations. And so I kind of feel that maybe from the investor side, there's a big box that's been ticked here. There's been lots of kind of unsuccessful bets. There's been lots of, for want of a better word, a lot of hype around novel checkpoints that hasn't materialized. And then we've kind of got, we've got one that, it's a well-controlled, well-designed study. But I think, when you speak to oncologists, that maybe that the response is, yeah, it's promising, but it's not a home run at this point. And obviously the thing that we haven't talked about is the fact that the Roche was studying the combination against Tecentriq as monotherapy, and that control arm didn't maybe perform to the level that KOLs would have expected it to, and there doesn't seem to be a kind of a particularly good reason other than, well, it was a small study, which kind of maybe does reiterate the point. Yes, this is promising, but yeah, we need to see the phase 3 data before we can make any kind of key conclusions.
Yeah, that was the extent of Roche's commentary there was, oh, this might have done better had we had more patients in it. It's a small effect. Mike, I don't know if your KOL had any divergent thoughts on that.
He said that he agreed that like the Tecentriq alone arm did underperform to a certain extent based on historical controls, but he also said, well, listen, I don't really care because I look at the totality of the picture here, and just what I saw from Tecentriq and tiragolumab, that combination looks very good versus, you know, historical or concurrent controls, I think was his quote. So he thinks that yes, the single arm did underperform, but he did not see that as taking away from, you know, the encouraged message that he took from the study.
Okay. Well, while we're on the case of the metastatic non-small cell lung cancer market, I just thought it was worth mentioning. We've obviously, you know, most of the focus was on TIGIT, but we've actually had a couple of recent approvals. The first approvals of BristolMyers Squibb's PD1 inhibitor and CTLA4 combination. So combining Opdivo and Yervoy. So it's been approved as a doublet IO therapy for first-line treatment of patients who are PD-L1 positive. So I think that's from kind of 1% expression levels upwards. That was in mid-May. And then actually just before ASCO took place, the FDA approved that combination with kind of a limited 2 cycles of chemotherapy. Kind of a slightly unusual situation because they approved the treatment and then included all the data that was going to be presented at ASCO in the label, but that showed a median overall survival benefit of 4.7 months versus chemotherapy. I mean, Michael, if you did the KOL you spoke to kind of have a view on where these treatments might be used?
Yeah, so there's a short answer and there's a little bit longer answer. The shorter answer is that he does not see this Checkmate 9LA regimen, which involves Opdivo plus Yervoy plus a limited course, meaning 2 cycles of chemotherapy. He doesn't see this changing his practice at all. So that's the short answer. The little bit longer answer is that if Bristol Myers is going to elbow its way into the frontline non-small lung cancer setting, he thinks it would be through the Opdivo Yervoy without chemotherapy regimen from the Checkpoint 227 study. But interestingly enough, he doesn't think it would be from the approved indication. So the approved indication, as you mentioned, was in PD-L1 positive patients. However, he thinks that the stronger argument, the stronger case for Bristol Myers would be in the PD-L1 negative patients based on the results from Checkmate 227. The issue is that was not like a agreed upon portion of the statistical design plan for that 227 study. So it's not going to be in the label. So this is really the longer answer here, that he thinks that perhaps Bristol Myers Squibb could use the data from Checkpoint 9LA and focus on the PD-L1 negative patients, and perhaps they could get a little bit of a market share that way. Other than that, though, the regimen itself, he just doesn't see it being a practice changer.
Yeah, I mean, it was interesting. You know, I think that there's always the question that there's the slight question mark with the doublet IO combination. That's Yervoy and Opdivo. Are you potentially, you know, I know in the past Bristol Myers has spoken about the longer term durability, and there's some kind of provisional data that speaks to the effect being quite durable, and you know, I think there's a scientific rationale for that, and obviously that will play out in time. It's interesting. I think that, you know, you speak to different people and there's kind of different ideas about this. There's that idea of, you know, you can kind of prime the immune system with some chemotherapy. You know, we know that immunotherapy works better when there's lower tumor burden. And, you know, someone sort of described the 9LA study as sort of trying to find the kind of the Goldilocks kind of, you know, not too much, not too little in terms of the chemotherapy element. But on the same, you know, talking about the same regimen, you know, you hear other KOLs say, well, actually, you know, basically to echo what you're saying, Michael, the person you spoke to, you know, some patients maybe incorrectly, because physicians know how to use chemotherapy effectively, but, you know, they'll take the opportunity to use a regimen that excludes chemotherapy. And the other argument I've seen as potentially supporting Opdivo and Yervoy is the idea that you're not using chemotherapy up front, you're maybe holding it back for patients who then relapse. So it's an interesting one, but I think we can go back and talk about one of the key themes in relation to the TIGIT data earlier. The results for Keytruda and the way that Keytruda is being used. It basically means that if any of these drugs are leaving have any kind of question marks around them, if the data is not kind of a home run, knocking the ball out of the park type affair, then they're just up against a therapy that has become very, very quickly entrenched. And like you said, hard to then shift treatment practice, I think.
Yep, I think that's a fair assessment.
Okay. Right, so let's move on to the last topic that we've selected. This is data from the Karthi study. This is looking at basically Karthi targeting BCMA in multiple myeloma. This is a relatively busy kind of late stage development space. I've seen people describe the J&J 4528 data that was presented this weekend as possibly the most exciting that they saw at the whole conference. I don't know if either of you two want to kind of, provide a bit of an overview of what we saw and where we kind of see things going.
Why don't you go ahead on this one, Mike?
Sure. Well, so, I think the takeaways for me were that number one, BB21, they provided an update from their, I guess you call it Karma study. No surprises. It just sort of looked like, it still looks good. They're still heading for approval eventually, perhaps, maybe. They recently got, they being Bristol-Myers and Bluebird, recently got a refusal to file letter from the FDA, which, is a bit of a setback, but they think that it'll be quickly addressable. And they also actually just got their application, I think, validated is the way it's the terminology in Europe. So they're on track for European approval. They think they'll be on a track or still on track for US approval soon. But the sort of the bigger, the highlight, if you will, from the anti-BCMA space at the meeting was definitely J&J and Legend's J&J 4528 program. So it seems to be the case that, the thinking seems to be crystallizing that this 4528 may be a little bit better than the Bluebird and BMS program. The efficacy looks a little better. Obviously, it's tough to say anything with cross-study comparisons, and this is, heavily pretreated patients. There's probably going to be some significant differences in terms of the baseline demographics. So it's hard to say too much on efficacy, but the efficacy does look, it still looks, and it has looked basically throughout, just slightly better for the J&J program. And then more importantly, perhaps, at least from a differentiation perspective, the safety profile looks more, basically the cytokine release looks like less severe, and perhaps most importantly of all, the cytokine release doesn't happen until basically like a week later. So it happens in almost every patient. I think it was 90-some% of patients will have some sort of cytokine release syndrome, but they only had a couple patients had grade 3 or 4, basically the real severe cases, and the median time to peak cytokine release was not until 7 days later. So the important part of that is that it would allow them to give 4528 as an outpatient for some or maybe even a lot of patients, which would be a big differentiation, a big point of differentiation against the BMS Bluebird product, which, it would need to be inpatient, and it'd be better on both convenience, allowing patients to get out of the hospital, go home, be more comfortable, and also on pricing, because right now at least inpatient pricing for CAR-T has been a big problem. I know that CMS is also trying to deal with that at the moment. Who knows if that'll go through or when it'll go through, but those are two big points of differentiation potentially coming away or basically being derived from 4528's better safety profile. So those were the takeaways from the BCMA universe for me anyway.
Yeah, and I would just expand on the efficacy bit a little bit, to the extent that we've seen this multiple times, whereas these BCMA targeting CAR-Ts release their initial data set, multiple iterations of these therapies have just come out on a first pass through the gate with an response rate of 90% and up. But of course, the issue here, is durability, where, BCMA and multiple myeloma, have a bigger problem with relapse relative to, some of the CD19 NHL CAR-Ts. So, it's really encouraging to me to look at these, duration of response graphs and see, how many of these patients are still in, sustained responses, at 9 months, after treatment.
Mm-hmm. Up to 11.5, I think, with the J&J. But yeah, that's a good point.
Michael, I think you touched on a really important point with the outpatient setting. It seems to be that we obviously a few years ago we had all this excitement around the CAR-Ts that were approaching the market and then subsequently came to the market, which target CD19. And then we've obviously seen the commercial realities, hit home, and to echo what you said, there's various challenges. So, it seems to be that the key question is, how do we get these in a point of care setting? It looks like, the combination of this efficacy, if it continues to perform as we've seen it, and this potential use in the outpatient setting, is a positive for the J&J and Legend compounds. Obviously, the other issue is this idea of streamlining the process with these allogeneic, off-the-shelf CAR-Ts. Becky, I know you were looking a bit at the Allogene data that was presented. What did that kind of tell us about how close to a reality these now are?
Right. So, from Allogene thus far, they've got, I want to say it's 22 patients that they've treated with their, most advanced candidate, which may not be their most advanced candidate for very long, as they sort of, go through iterations of re-engineering their cell therapies. But the good news for Allogene with their CD19 targeting CAR-T for NHL is they have, sort of hit the minimum barrier to entry, I guess, that they needed to at this point in time. They have, they've seen no cases of graft versus host disease, which is probably, the biggest theoretical concern for transitioning over to this kind of universal donor type cell therapy. And they have what they're describing as manageable rates of cytokine release syndrome. So while the bigger question for them, knowing that their efficacy, is sort of in the same ballpark, they're not embarrassingly behind, the autologous therapies in terms of response rates or anything. But the big outstanding question to sort of figure out whether or not we can use these allogeneic therapies, as a standalone treatment, or as sort of a bridge to transplant or bridge to autologous CAR-T, is what the persistence of these therapies looks like. And this is, still going to be a burning question for some time yet. I think Allogene is looking at about, 3 or 4 months of progression-free survival yet. So we'll need more kind of extensive follow-up data to see if, that metric is comparable to the autologous counterparts. And that will be sort of, I think, a big deciding factor in where they end up fitting into the treatment landscape.
Okay. Well, it's certainly, I think we've we had a lot of excitement around Karthi, and then it's kind of, you know, dipped, but it looks like it's going to kind of pick up again. I think, you know, I think certainly in the short term, hopefully with these BCMA targeting agents in multiple myeloma, but obviously the allogeneic stuff as well is, you know, it's going to be fascinating to sort of see how that evolves and whether the potential is met. Thanks, Michael. Thanks, Becky, for your time this afternoon. Thank you to everyone who's watched this, who's listened to it. There was obviously lots of other interesting data presented at ASCO. There's lots of coverage on FirstWordPharma.com. So just obviously kind of, you know, make sure that you check our other coverage out. And we'll speak to you again soon. So thanks for listening.
Cheers.